Thank you for the introduction. As Bas said, my name is Michiel van der Flier. I'm a Dutch pediatrician. You can all hear me well? Yeah, great. Um, Thank you for the invitation. I was asked to talk about infections and immunity in chromosome 11 diseases. Next slide, please. Oh, I can do that, of course. Sorry. I'm a pediatrician based in the Netherlands in the Children's Hospital in Utrecht. And uh, I see children with infectious diseases in the hospital, but also children with immune deficiencies, and including syndromal immune deficiencies like Jacobsen Syndrome. And I'm happy to be able to tell a bit. I'd like to start by introducing the immune system to make it easier to explain what's specific about Jacobsen syndrome. This is a screen the older among you may recognize. It's one of the first computer games, Pac-Man, and it was very popular when I was a youth. And Pac-Man is the yellow um, um, circle in the bottom of the screen, eating white dots. And actually, this looks very much like one of the cornerstones of our host defense against bacteria, the phagocytes. Phagocytes are cells that are literally called eating cells in Latin, and they eat bacteria or rubbish they find inside of the body. So though we are covered with bacteria on the surfaces of our body and in the gut and in the airways, which is normal, they shouldn't enter the tissues. And that's when phagocytes will come into play and eat them and kill them. This you all recognize as a hamburger. It's a metaphor for a bacterium being eaten by a phagocyte. And this is what regular bacteria look like to phagocytes um, without additional help from the immune system. Not very attractive to eat. If you add some lettuce and cheese and sauce, it will become much more attractive to eat. And it's the same way for bacteria and phagocytes. Bacteria need to be covered with opsonins, antibodies, complement immune molecules of the body to make it easier for the phagocytes to eat them. So when we talk about immunoglobulin deficiency in Jacobsen's syndrome, you can compare it to an empty bottle of ketchup. And you think back of the hamburger, which isn't very attractive without any additions. And you can see why the body wouldn't be efficient in clearing bacteria entering the body. This whole process of bacteria being covered by antibodies and other immune molecules like complement, making it easier for the phagocyte to eat them, digest them, and kill them, and eliminate them from the body is called opsonophagocytosis. So we already touched on one immune deficiency, a lack of antibody, immunoglobulin deficiency, but defects may also arise in the phagocytes themselves, which are called also neutrophils. We all know if we want to clean a place from germs, from bacteria, um, bleach is very effective. Hydrochloride acid, chlorine. And that's exactly what's in the inside of these cells. So if a neutrophil, a phagocyte, eats a bacterium in its interior, in its stomach, the bacteria is killed by bleach. And the bleach inside the bacterium also activates enzymes that cut up proteins of the bacterium to kill them, uh, proteases. Um, but we know there's some diseases where this process is malfunctioning, and they are called phagocyte functional defects. And this is another issue that may occur also in Jacobsen's syndrome. There's other diseases where there's just a shortage in number of neutrophils, and that's called neutropenia. That's an important immune deficiency, but it's not at play in Jacobsen's disease. But we see that, for instance, in patients with cancer. If they receive chemotherapy, then they will become neutropenic and prone to infection. Neutrophils and phagocytes aren't the only immune cells that are important for your host defense. Um, they are very important to guard off bacteria, but for viruses, we need other defenses. And viruses, when they infect us, start to live inside cells of our own body and use the machinery of the cell to multiply themselves. And the surfaces of our cells are made of a membrane made by lipids, which is fat. And you can compare the fluidity of the lipid membrane of cells with the um, fat bubbles you see on chicken soup broth. What we all know is when the soup is finished and we do the dishes and we use soap, that gets rid of the lipids, of the fat. It disrupts the lipid membranes. And this is something that our own host defense also uses. Our lymphocytes contain molecules that look like soap. And when we have a cell infected by viruses, it's recognized, and our lymphocytes can bind that cell and excrete the soap, disrupting the membrane, shooting holes in the membrane of the infected cell and killing it. So the virus is stopped multiplying. Here you see an electron microscopic picture of a lymphocyte attacking a virus-infected cell. The lymphocytes on the left of the screen and the virus-infected cell on the right. And the cell sort of kisses the other cell, forms an immune synapse, and excretes all these soap-like molecules that lyse the virus-infected cell to stop the virus. And what's important to mention in that is that this is the work of some lymphocytes, and there's other lymphocytes that produce the immunoglobulins that we talked about earlier. So lymphocytes are of different types. So when you look at the summary of our normal immunity, a normal host defense, you see on top in red the phagocytes that can eat bacteria. They are called innate immunity or inborn immunity. And at the bottom, you see the lymphocytes, which are T and B cells, and they shape the acquired or adaptive immunity. And they learn to recognize specific pathogens. So T cells are in different kinds, and each T cell recognizes a specific virus or a specific bacterium. And the T cells attack infected cells with soap, and the B cells produce antibodies against that specific virus or specific bacterium to enable opsonophagocytosis by the phagocytes to kill the bacteria. And the process is multi-layered, so the antibodies also activate complement, which helps opsonize bacteria and may also lyse cells and bacteria. So there's some redundancy in the system. If you look at our body, um, we know the heart and the blood vessels, but in parallel to the blood vessels, there's also a whole vessel system that's part of the immune system, the lymph system. And for the people in the room that have been in Holland, we have bike lanes attached to most roads. So you have the main road, you can compare that to the blood vessel where the red blood cells travel. And parallel to that, you have the bike lanes which you can compare to the lymph vessels in the body, where leukocytes travel and ingested pathogens. And we all know the tonsils and the adenoid, but we have also, when we have an infection, um, a lymph node may enlarge in your um, neck in response. And that's caused by multiplication of the immune cells in that lymph node, responding to an infectious threat. And in the figure, you see depicted in the chest the thymus, and that's a lymphoid organ which is comparable to a school. This is where the lymphocytes go to school, and when they graduate, they are T cells, recognizing a specific virus or bacterium, and when it infects, they can help attacking it. You also see in the belly the spleen depicted, and that's like a T-sieve. It sifts the blood all the time and sifts out invading bacteria. And the spleen is full of phagocytes to directly eat and digest bacteria that have entered the bloodstream. You see all the other lymph nodes depicted, the appendix, which can be inflamed, as you all know, and sometimes you may need surgery to remove it. is also part of the immune system, of the lymphatic system. And the bone marrow, which is the inside of the bones, is sort of the factory where all the blood cells are produced. So that's where both the red cells, the thrombocytes, but also the leukocytes, the immune cells, are produced. So in normal immunity, if you zoom in and you look at the B cells, the B lymphocytes, that are the cells that produce immunoglobulins or antibodies that recognize a specific virus or bacterium. So you see they are different. That's why they have different colors. And the immunoglobulins they make are also different, recognizing different um, uh, pathogens. And that's why they also are depicted in different colors. And if you look at the pathogens, you see, for instance, the yellow one will only be recognized by immunoglobulins that are yellow, produced by the yellow B cell. So it's a very specific system. And once it's activated, the cells will multiply and start producing a lot of antibodies to help defend the body against the infection. So we already touched on antibody deficiencies and immune deficiencies. So think back of the ketchup and the hamburger. If there's something wrong with the B cells and they don't mature or they can't produce antibodies, you have a lack of antibodies and are more prone to infection because opsonophagocytosis doesn't go right. There are several diseases like X-linked A gamma globulinemia where there's no antibodies at all. And there's a lot of diseases that have a partial deficiency of antibodies, which is what also may happen in Jacobsen syndrome. When we speak about antibodies or immunoglobulins, it's good to realize that there's different types. They have different letters to denominate them, and you see the little drawings of the molecules that they look different. There's IgG, that's in the bloodstream. It's present in the bloodstream and it's the most crucial one to defend you against bacterial infection. There's IgA, which is located on the mucosal surfaces of the body and also in the bloodstream. There's IgM, which is produced very early in a new infection when there's a new pathogen encountered. It's present in the blood and also transported to mucosal surfaces. There's IgD, which we don't know why we have it. And there's IgE, which we know is very important in allergy and in infection with parasites like worms. And you may see that different classes of immunoglobulins are missing in a single patient. And when we think of the pathogens that surround us, there's fungus. You see on the left top panel fungus in a petri dish in the laboratory, but you may also see it on garden rubbish or on food that has gone bad. Fungal pathogens form spores that survive for very long times and may start growing when the conditions are right. So we all are colonized by fungal spores, which our immune system keeps in check. But if we become immune compromised, fungi may grab their chances. And what we know is that, for instance, young babies whose immune system hasn't matured yet are prone to fungal infection. They can have thrush, the white plaques in their mouth, and that's a yeast infection, a fungal infection. also people with a normal immune system may have a fungal infection on their skin, a dermatomycosis. On the bottom, on the left, you see bacterial cultures in the petri dish in the lab. And bacteria are present on our mucosal surfaces, in our gut, in our poo, um, on our skin. And yeah, we all know that they frequently may cause infections like pneumonia or like bloodstream infections, sepsis, or bone infections, osteomyelitis, all sorts of infections. And on the top right panel, you see different viruses depicted on scale. So some are small and some are quite big for a virus, but still a lot smaller than a human cell. And the virus, the second on the right at the top, is probably the shape you're most familiar with, because that's also what the coronavirus looks like. And we've all seen a lot of pictures of coronaviruses in the media. But the one on the most right, for instance, is the shape of a snake, and that's what an Ebola virus looks like. So different viruses have different shapes. And all these pathogens can be transferred from one person to another. And what we all have experienced that during Corona lockdown, with the social distancing that was happening, common colds and most infections massively decreased in numbers. And for some patients with immune deficiency, it was actually quite a good time in that sense, that they suffered far less infections. But the lesson we can take with us in current times is that um, social hygiene can help prevent infections, so that in some moments you yeah, can take some basic measures to help prevent infections. What we've learned in the past decades is that we know diseases that are primary immune deficiencies, inborn diseases with a defect in one gene that causes an immune deficiency. And I mentioned XLA, X-linked agammaglobulinemia, where there's one gene defect, B cells don't mature, and you don't uh, have the capability to produce immunoglobulins at all. That's a clear example. We also know there's what we call secondary immune deficiencies. When you use a drug that inhibits your or hampers your immune system, like chemotherapy and cancer, you also become immune deficient and prone to infection. But we've also seen that children and people with syndromal conditions, where there's complex genetic mistakes, and you can think of Down syndrome, where there's an extra copy of the chromosome 21, or you can think of the Jor syndrome, where there's not in chromosome 11, but in chromosome 22, there's a deletion of part of the chromosome. But there's a lot more diseases where a chromosome misses a little part or has an additional part. And as a result, you see different symptoms in a patient grouped together and recognized as a syndrome. We have learned that as part of these syndromes, you can also see immune deficiency. And a few years ago, we did a survey in Europe, and through the Immunodeficiency Society, 16 hospitals cooperated to look in their populations. And we had in these 16 hospitals, there were 46 patients with immune deficiency that was part of a syndromal condition. And 4 of these were patients with Jacobsen syndrome. So to give some impression of the um, importance of Jacobsen syndrome as a cause of syndromal immune deficiency. So about 10% of the syndromal immune deficiency was caused by Jacobsen syndrome. That excludes children with Down syndrome and the 22q11 deletion syndrome. But it's an important group of children and adults. So if you think of the people part in the network that have a chromosome 11 defect, when should you test the immune system, or when should you think about the possibility of immune deficiency? Well, if you think about the immunoglobulin deficiencies, they mostly present with respiratory tract infections. And that can be acute otitis media or sinusitis or frequent pneumonias or lower respiratory tract infections. If you are unlucky, you can also suffer a much more severe bacterial infection like sepsis or a meningitis. And if you think about the Jacobsen associated immune deficiency, it's not only antibody deficiency, it's also lymphocyte numbers that may be too low. And that makes you susceptible to viral infections. So you may also have chronic diarrhea. And what also can happen is fungal, like the little baby, fungal skin and mucosal infection. So as an older child or adult, you may develop thrush, which isn't normal beyond the neonatal age. So if we want to test the immune system, we have to take some bloods. And to simplify it, what we do is count all the different immune cells to know if they are all present in the correct numbers. And we also can measure the amount of immunoglobulins. Sometimes we simulate an infection. We give a vaccination and assess the production of antibodies in response to that vaccine. It's also possible to see if the lymphocytes can multiply properly. But in general, that doesn't help us very much to come to a diagnosis. So the counts of the cells are in general most important. So if in a child with frequent acute otitis media, you see that the immunoglobulins are low, combined with Jacobsen syndrome, it's likely that there's really a true immune deficiency associated with the syndrome. And the same applies to frequent viral infections. If you have very low lymphocyte counts, that's a likely explanation why you're prone to have these frequent infections. Why does chromosome 11 disease cause immune deficiency? This is a picture I suspect many of you have seen before. If you think of a human cell, we have this sort of this football-shaped structure in mind in general. Cells can have very different shapes, but think of it as a football. And inside there's a little ball which is called the nucleus. And in the nucleus, the chromosomes are stored. And humans have 46 chromosomes, 23 pairs of identical chromosomes, one from the dad, one from the mom. And what you see here is a technique to visualize the chromosomes in a human cell. It's called a karyogram. And if the chromosomes are photographed and cut out, you can pair them. And what you see is there's a little arrow at the chromosome 11 pair. And what you can see is that one of the two is a little shorter than the other. And the lack of that little piece of DNA is the cause of Jacobsen syndrome or chromosome 11 deletion disease. And in this little piece of DNA that's lacking, multiple genes are located. And in general, a gene encodes a protein. And the DNA is read through your body and serves as a building plan for proteins. And the proteins are either building blocks of your body or they have a function to, for instance, as an enzyme, catalyze chemical reactions to produce other metabolites and help the machinery of our body to work. So to simplify it, if one protein encoded by one gene is missing, you still have another copy of that protein on the other chromosome. But for some genes, missing one copy is already deleterious. On the part of chromosome 11 that's missing, there's a number from 6 to perhaps 20 or more genes, depending on how big the part is that's missing in a specific person. And of course, we are interested to know what genes on that piece of DNA are responsible for the immune deficiency, because if we know, we can check and predict who we have to monitor for immune deficiency or who doesn't miss the gene that determines this and is not at risk of immune deficiency. There's a few genes that have been thought about to be logical candidates to cause the immune deficiency. And one was FLI1 and the other was ETS1. And what you see here is a diagram where you see that different inflammatory stimuli like interferon or endotoxin from bacteria stimulates the production of this gene product, and that the gene product opens up the DNA to promote other genes to be read and other substances, proteins to be made. In other words, these are genes that encode transcription factors that regulate protein production from genes. And what you see here is that if the gene is stimulated, many cytokines or chemokines are all inflammatory substances are produced. So it's logical that this would have a role in the immune deficiency if this gene is not present. So this may still be true, but biology is complex. And it turns out there's also patients around that have immune deficiency, miss part of chromosome 11, but still have these two genes. So it doesn't explain everything, which makes it hard to grasp. What you see here is a map of the three-dimensional structure, how the chromosomes are folded up in the nucleus of a human cell. It's actually quite pretty. And what we've learned is that in all humans, the chromosomes are packed together in exactly the same way. So it's not random. And the DNA is folded up very neatly. It's like a rope around a little notch that you wind it around to store it so it doesn't get mixed up. So the DNA is also wrapped up neatly and packed very orderly in the nucleus in the cell. And if a part needs to be read, that specific part will unwind and it can be read and it will be packed up again. So the genes we mentioned previously are involved in the unpacking of parts of DNA so that it can be read and transcripted and proteins can be made. And what we've learned is that if a piece of chromosome is missing, this also influences the reading and unwrapping of genes in other chromosomes that are laying adjacent in the 3D structure in the nucleus. So missing a part of DNA can have broader effects than just the genes that you lack in that piece of DNA, which makes it very complex. So we still don't understand a lot of it, to be frank. And the positive thing is that there's luckily a lot of redundancy in function. So a lot of things can be um, all right, even if you miss certain genes. But you do see that there's less efficiency in functions, and you do see the consequences of that. And focusing on the immune system, if you look at the general population, the chances of a person being diagnosed with common variable immune deficiency, which is antibody deficiency in two of the classes of immunoglobulins, say IgG and IgA or IgG and IgM deficiency, plus frequent infections or insufficient responses to vaccination. That's sort of the definition. That's 1 in 25,000 individuals. So that's a very low number. It's more frequent than Jacobsen syndrome as such, but it's still very infrequent. So if you look at a country like the Netherlands, there will be 6 children born with CVID each year, and there will be 2 children born with Jacobsen syndrome. So it's rare. Inside the population of people with Jacobsen syndrome, the frequency of antibody deficiency is much higher. It's really not possible on the available data to give an exact number, but it's a lot higher. I don't know. It's because the cohorts are biased to cohorts from immunologists. It's hard to say the real number, but yeah, I would say at least a third, um, to even more than that, will have immune deficiency. And when to check? I think if you have such a high risk of immune deficiency, it's defendable to test everyone to see if their lymphocyte numbers are okay, to see if their immunoglobulin levels are okay, and to do that every so many times to monitor if it stays all right. If you have a lot of infections, recurrent or severe infections, that's a direct indication to check in the um, Jacobsen group where you have such a high risk of immune deficiency. And I don't know if we have enough data to say really like you have to check every so many months. I think there's a human factor involved too. So I think that's something you have to decide on an individual basis. If we check, what can we do with that information? Is it useful to know? I think it is, partly. Of course, if you know that you are susceptible to infections, you would like to prevent those infections. And if you become ill with an infectious disease, you know you have to be more on top of it to treat it right. So that's the gain of knowing there's immune deficiency. And one thing is important is optimal vaccination. Even if you have antibody deficiency, vaccination also has an effect on the lymphocytes, on the T cells. So vaccination may still be effective. So that's important, vaccination. If you think about viral diseases, for instance, flu, um, you may be more prone to flu if you have lower lymphocytes due to a lymphocyte deficiency, and a flu jab would be recommended. If you have a mild antibody deficiency, say IgA or a little low IgG, then antibiotic prophylaxis may help reduce the number of ear infections or chest infections. So antibiotic prophylaxis can be crucial. And we don't want all children to use antibiotics because then we will get massive resistance. None of the bugs will be responding to our drugs anymore. So we have to be uh, selective in using and prescribing antibiotic prophylaxis. But if you have a mild antibody deficiency, that's a accepted indication, and the individual benefit outweighs the societal risk. If you have more severe antibody deficiency, you can fix that by treating with immunoglobulin suppletion therapy. if there's too little antibody, we can use immunoglobulins from blood donors to fill the gap. And it's only IgG we can give as a infusion. There's at present no IgM or IgA therapy. So it's just IgG, but it is the most important to prevent severe infections. And patients receive infusions with immunoglobulins either by an intravenous drip or by a subcutaneous infusion. And it can be weekly or monthly. It depends on the preparation and on the circumstances. And often it even can be given at home. Also depends on the local circumstances. But in a lot of countries, you can do home therapy and are independent of nurses and can supplement the antibodies to a normal level. And the aim is to reduce the frequency of infections and to prevent severe infections like sepsis or meningitis by the antibody therapy. And some of you may have wondered, if you have a deficit of antibodies, how can a vaccine work then? Because the vaccine generally works by inducing antibodies against the pathogen. But there's two answers to that. The first is also the lymphocytes are stimulated by the vaccine and may help fight the infection. And when a patient receives immunoglobulin suppletion therapy, they will also get antibodies against all the pathogens that the population is vaccinated against through the antibodies from the donors. So the immunoglobulin infusions also help protect against viral infections like measles, etc. There's a risk with all donor-derived blood products because in the past we have learned that there can be viruses transmitted through blood products from donors to patients, like HIV or hepatitis B, as has happened in the past. But with the current strict guidelines, HIV and hepatitis B are not transmitted anymore because the procedures are safe. So there's a theoretical risk of a new bloodborne pathogen that could be transmitted, as we've also seen corona appear. You can never completely exclude that, but to all standards, it's currently safe. I'm at the end of my presentation. I knew with the live translation, I would have to speak slowly, but I had no idea how long it would take. So I covered sort of the general aspects of the immune system, some specific about the mechanisms why in chromosome 11 disease we see immune deficiency, and some aspects of the treatment. But there's room for questions or discussion or experiences to share. So I'm happy to hear. I missed the last part of the question. I'm sorry. Right. Yeah. Yeah. That's a very good question. Because indeed it does. And if you have immune humoral immune deficiency, so you lack antibodies, um, often also the mucosal antibodies will be deficient, and they are one important part of the gut defense. And what you see is an increased incidence of, for instance, chlamydia, no, not chlamydia, giardia, gut infections, which is a parasite. Um, so there are some centers that routinely screen all patients that have hypogammaglobulinemia on giardiasis, doing fecal tests. What we usually do is that we test if there are complaints, but it's a good addition. So I focused on viruses and bacteria, but you're absolutely right that also gut parasites have an opening if the antibodies are low. And you don't resolve that with IgG therapy because we still lack the IgA, which is so important for the mucosal protection. Does that answer your question? Yeah, that's important you mention it because warts are caused by specific viruses, the papilloma viruses, and like other viruses, host defense against these viruses is through the lymphocytes. The cytotoxic T lymphocytes have, in the end, to eliminate the papilloma virus infected cells. In healthy children, warts can be quite persistent, and some children have warts for years. But in children with low numbers of T cells, this can be excessive. And it can really cause a lot of problems mechanically, cosmetically, and it can be very persistent. And um, this is a very difficult problem because we don't have drugs that sort of fix the T cells to do this. And if there's immune deficiencies with really life-threatening uh, severity of T cell lack, we go to very aggressive therapies like stem cell transplants, and then the children get a whole new immune system, and the T cells are part of that new immune system, and the warts will disappear. But for not life-threatening immune deficiencies, but with this wart problem, it's really problematic. Um, Personally, I'm not aware of T cell therapies in Jacobsen patients, but I thought you mentioned you knew some children in France had received this. Am I correct?
No, I don't know.
No, no. So I think it's something we're all struggling with because there is no easy fix. Um, think there's perhaps an outlook to the future because there's much progress in engineering of cell therapy, which is now used for cancer and is very expensive. Um, but this might be applicable to infections like this. And it may be possible in the future to target T cells in the lab against HPV, the papillomavirus, to treat this kind of warts. But at present, I don't think there's studies ongoing testing that. Um, but it's absolutely one of the values of a network like yours, the chromosome 11 European network, that um, we bring all the people together, and we also can hear from um, uh, um, treating clinicians of all these persons. And if someone somewhere thinks of something smart, it can help other people. But I'm afraid at this point, this is one of the immune deficient symptoms where we really lack a good solution. Sorry. Can I just ask, is there more people in the room that have these ward problems? I hear quite a number of yeses. Yeah, so it's a significant problem. Yeah, yeah, that's difficult. Yeah, that's a very relevant question, and I stepped over it in my presentation to simplify, but it's important to explain. Um, I'm just thinking. Yeah, I'm going back a few slides to illustrate it. Um, if you think that what you see here is not B cells, but think a T where the B's are. So these are all T cells. And say the little dots are not pathogens, but they are virus infected cells. Then each T cell can only recognize cells infected with a specific virus. You need a whole range of different T cells that have gone to school in the thymus to learn to recognize that specific um, structure or pattern. When this works through receptors on the cell, and it's the T cell receptor that recognizes little pieces of this virus on the surface of infected cells. And this is very important in viral defense, but it needs a little time to get activated. And because there's only one T cell that recognizes that specific virus, it needs to multiply before it has enough force to eliminate all infected virus cells. And nature has thought of a sort of backup mechanism, and it has also produced lymphocytes that are part of the innate immune system, like the phagocytes. And the innate immune system doesn't need to go to school. So the receptors on these innate immune cells uh, recognize very broad changes on host cells that are signaling they have changed and have fallen ill. And that can be general patterns that are present in a tumor cell or in a virus infected cell. Things change on the surface, and the innate receptors recognize that cell is off. And the network killer cells are the lymphocytes that have those receptors. They don't have T cell receptors, they are not part of the adaptive immune system, they are innate immune cells. They recognize a cell is strange, but they have the same soap to lyse that cell. So they also make an immune synapse, secrete perforins and other toxins that shoot holes in the membrane of the cell and kill the virus infected cells. So they are a backup system for the specific lymphocytes, but the function in the end is quite the same. Now, in Jacobsen syndrome, but also in other immune deficiencies, we know there are individuals that lack NK cells, and some have no complaints at all, probably because their lymphocytes cover the hole. But some have, for instance, inflammatory bowel disease or other uh, immune problems or viral infections. Because it is one of the defenses against viral infections. It's hard um, to solve, but that's sort of and your daughter, she has viral infections, or yeah, yeah. So it's the preventions you can take are limited. I mean, you can use the uh, and she has low NK cells. Then you hope that there's still a reasonable number of T lymphocytes. That would help. Um, you can give the flu jab to try and prevent influenza. Um, but for the other viruses, the respiratory viruses, we don't have any preventive measures. We can sometimes, if the immunoglobulins are low, that can help a little bit. But for respiratory viral infections, immunoglobulins usually have a minor effect. It's more important for the bacterial pneumonias. So it has some effect, but usually not massive. And of course, antibiotics can't prevent the viruses from infecting. So we are a bit stuck there, unfortunately. So it's difficult to solve. Right? Yes. Yeah. Well, that's yeah. So I missed the indication for the removal of the thymus. Did you say cancer or heart surgery? Cardiac surgery. Yeah. 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 So you're right. Cardiac surgery is a frequent um, uh, cause of loss of thymus, um, and yeah, it's depending on the local sort of the situation the surgeon arrives at, if it's possible to keep some of the thymus or whether it's really hampering the surgery. Um, there's also inborn diseases where the thymus is not um, formed correctly. and that's with another chromosomal deletion syndrome, the 22q11 deletion, the DiGeorge syndrome, where um, genetically the generation of the thymus doesn't occur. Um, in those children, currently trials are ongoing to see if thymus transplantation can help. And that's a fascinating idea, to give thymus from post-mortem donors to a patient and to see if that can teach the lymphocytes that are present to become better functioning T cells. And practically what they do is they inject the thymus parts in the um, abdominal cavity, in the peritoneal cavity, so in the belly, and they seed there on the tissue and become connected with blood vessels and start functioning, but it's still quite experimentally. But this might be something that might have application in the future also to other patient groups. So um, uh, this is something to watch for with your uh, clinician, if that might be something of interest to your daughter. But it's still quite experimental, and I think it presently is only done in London. Yeah, that's a good question. And so there's a few aspects to this. Um, even in a immune system that's abnormal, there may be some maturation. So some things may improve in time. Um, and um, as there's also a lot of viral infections ongoing, the immunoglobulins aren't the only determinant of respiratory infections, because they won't be all bacterial. And with increasing age, you generally get less viral respiratory infections. So that may help. And the antibiotics are intended to decrease the number of infections and the severity. Um, what is most important is the absolute value of the immunoglobulins. So normal adults and older children have above 7 grams per liter immunoglobulin G in their blood, and that can be up to 15 gram per liter. If you have a immune deficiency, if you have hypogammaglobulinemia, it may be that you are, say, having 5 gram per liter, and that may be enough. If you have, for instance, only 3 grams, we know by experience that there's a really serious chance of a severe infection occurring. So that's something we usually weigh in the decision. And it may also be that it's possible to test the quality of the antibody response. So that can be done by giving this vaccination to see how the immune system responds. And if you do that and you see a perfectly well response, though the levels are a bit on the low side, that may also help estimating that it's safe not to give the antibiotic prophylaxis. One other thing is that it's difficult to take lifelong medication. We also see that with patients that don't have a spleen, the risk of infection stays lifelong. But to take preventive antibiotics every day, lifelong, is very hard. It ends up not being taken. So in that sort of situations, it can also be an alternative to say, if you fall ill, start antibiotics directly. And that may also be a safe strategy.
Yes, and perhaps a follow-up question. This testing with the vaccine that you mentioned, would it be useful to repeat that now that she has grown a bit older? Because I know it was done when she was younger, but that's been several years now.
Yeah, so you can we sometimes repeat it, say after 5 years or longer, to reevaluate. Yeah, so that can be done.
Okay, thank you. Any other questions? If not, then let's put our hands together once more. Thanks so much for your digital presence. I really only asked Dr. van der Flier roughly a week ago, and he made time in his weekend. So thanks a lot.
You're welcome.
And well, hopefully we can stay in touch as the network with you.
You're very welcome.
Thanks.
Well, I hope you have an enjoyable sequence. Okay,
thank you.
Bye-bye.
Bye-bye. So that was immunology.